a particular compound for a particular patient. And when we talk about individualization of diabetes therapy, I think we have quite a number of tools at our disposal at the moment, but we don't necessarily have all the tools. And there are certainly safety considerations that need to be exercised. I think you're all aware, and we've just been reminded of this, that presently there is something like 387 million diabetics across the world, particularly focusing on type 2 diabetes here, and this number is expected to rise quite significantly. This is a massive burden on healthcare services across the world, not only in the Western countries, but also in developing countries. So this puts huge pressure on public resources. From a patient perspective, the consequence of diabetes is really cardiovascular death, which is increased twofold for diabetic patients, and very significantly in terms of all-cause mortality as well. We talk about cardiovascular mortality, but actually the burden of the diabetic disease is due to multiple abnormalities that the patients go through, ranging from being associated with hypertension, being associated with chronic kidney disease, being associated with dyslipidemia. And as Claire just pointed out, there is increasing evidence that there is a significant role for sympathetic nervous system activity and brain involvement in the regulation of metabolism, and the fact that insulin resistance causes significant dysregulation of these pathways. This multiplicity of effects in multiple organs effectively results in damage to various tissues, resulting, as we just talked about, in increasing heart attacks. There is also a significant increase in eye abnormalities, diabetic foot ulcers, nephropathy, neuropathy, and of course stroke. So really the aim of treatment is to treat ultimately the complications, to reduce the complications of the disease as far as is possible. The tools that have been available for the past years have really targeted glucose lowering by generally stimulation of insulin secretion. And this is data from the UK PDS study, where it was shown that despite the inexorable decline of pancreatic function and the worsening of glycemic control, it could be shown that diabetes-related endpoints, if the patients were treated aggressively, were certainly improved. And in particular, microvascular complications, those affecting the eye and kidney function to some extent, are also improved as a result of this intensive treatment. What was rather surprising at the time was the fact that although microvascular complications were beneficially affected, macrovascular complications and certainly diabetes-related death was not improved whatsoever, despite this improvement in temporary improvement, at least in glycemic control. So because at the time the majority of patients were on sulfonylureas and or metformin treatment, the pharma industry was looking at identifying possible new mechanisms to tackle the disease. And as Claire pointed out, there are two sides to the coin. One is abnormalities in insulin secretion, and the other one is insulin resistance that patients suffer from. So many companies started looking at trying to tackle insulin resistance. If we look back from the history of of diabetes treatments, therefore, as I just pointed out, most of the treatments in the 50s, 60s, 70s were due to SUs and metformin treatment. And the others that we've enjoyed the experience of have been glucosidase inhibitors that have generally low efficacy in terms of glucose reduction. Human insulin, of course, recombinant human insulin has changed the field. But what I'd like to focus a little bit on is glitazones and particularly their insulin sensitizing action. In fact, as Andrew and Claire pointed out, the identification of rosiglitazone in particular was spurred by a Japanese publication which showed a predecessor compound, siglitazone, which was being developed as an anti-hyperlipidemic agent, also showed some glucose lowering effects. So in those days, and I'm talking about the early 80s to 90s, really the pharmaceutical industry was doing bucket biochemistry, so using large test tubes and testing compounds in animals. They decided that at Beecham Pharmaceuticals in those days to start modifying the siglitazone molecule in order to identify compounds that had better glucose lowering effect. They weren't particularly interested whether they maintained the hyperlipidemic effect, hypolipidemic effect or not. And the testing was done in rodents in order to demonstrate glucose lowering effects. At the time, it was absolutely not known what the target of these glitazone molecules is. We, of course, now know that these are p-power gamma activators, these glitazones. But at the bottom of the chart here, I just wanted to show that we've moved away very much from using what I refer to as bucket biochemistry to using cells and cell lines. And we now do, in the pharma industry, high throughput screening, so 384 well plates, even 1256 plates, in order to rapidly test multiple compounds that chemistry colleagues generate. They have many chemical libraries and targeted libraries that they can generate, and this allows the pharmaceutical industry to effectively test hundreds of thousands of compounds within a relatively short time period in order to determine which ones are more potent, which ones have interesting characteristics. Of course, they still need to be put back into animals in order to demonstrate efficacy in vivo and potential side effects. But nevertheless, the advent of these technologies has effectively resulted in the development of many more classes. If you compare that between 1950s and 1980s, we really just had 3 classes of antidiabetics, metformin being the sole member of each class, compared to what we've experienced in the last few years. It's just a testimony to the success of this. Although you do hear a lot about this in the news, that the cost of this, of course, cost of drug discovery has increased because of these technologies. Nevertheless, it has been associated with success, in my view. Just going back to the rosiglitazone and the p-power gamma agonists, it was in the late 90s that the first of these compounds was approved, troglitazone, which unfortunately was removed 2 or 3 years later due to liver toxicity, which was identified in the clinic. In the intervening years, 2 additional compounds, Rosi and Pio, were also approved by the FDA and also subsequently in Europe. And one of the initial concerns with both of these compounds that the regulators had and the clinicians had was, of course, whether these were also going to have the liver toxicity that troglitazone exhibited, and the companies were requested basically to perform trials in order to demonstrate safety, which by and large has been the case. So the profile with respect to liver toxicity of Pio and Rosi is very, very different to troglitazone. It does not cause the toxicity that TRO exhibits. As was mentioned, the holy grail in diabetes treatment is really improvement of cardiovascular risk-benefit, as had been indicated by the UK PDS as well. So both companies involved in the development of these agents were running cardiovascular trials, and one of which was Proactive, which I think Bob is going to refer to quite significantly later. So I'm not going to touch on that at all. But just to say, in terms of rosiglitazone, what had happened is that a number of other issues came to light with both of these compounds. In particular, possible risk of heart attack with rosiglitazone, which was shown in a meta-analysis of 42 trials. And this effectively resulted in the removal of rosiglitazone from both European and the significant restriction of its use in the US. Pioglitazone continued to be on the market. It still continues to be on the market, but it has been suggested that it is associated in some patients with possibility of bladder cancer. And certainly there are cautionary notes that you see in the literature saying that people who may have had bladder cancer should not be prescribed pioglitazone as a precaution. Despite this withdrawal of rosiglitazone from the market, there were one or two long-term studies that were going, and they were actually ongoing, and subsequent cohort analysis of these by FDA has resulted, in fact, in the total removal of the restrictions that rosiglitazone had been subjected to a few years earlier. In the intervening years, the company, GSK, I think paid out something like $3.5 billion of lawsuits to patients and to regulators. So there is a lesson to be learned here, I think, with respect to making sure the cardiovascular safety of drugs is addressed early on, certainly. And in fact, there was another story that was going on in the minds of FDA as this rosiglitazone cardiovascular saga was continuing. I already touched upon troglitazone, so I'm not going to go through that. But by 2006, this is still a couple of years prior to the rosiglitazone cardiovascular story, the FDA had been through 50 additional INDs for various ligands of the PPARs from different pharmaceutical companies. Everyone wanted to tackle insulin resistance as the side of the coin of the disease that had not been attacked. And all of these PPAR ligands had been terminated due to safety concerns. So some of this was due to cardiovascular, some of this was due to cancer, some of them due to other issues. And in fact, even prior to the requirement of cardiovascular safety studies for anti-diabetic agents as a whole, the FDA had requested that there should be cardiac safety from chronic preclinical and phase 2 studies, plus submission of 2-year rodent carcinogenicity data before a PPAR compound could go into phase 3 development. So I guess the clinical data and the meta-analysis that came through basically were the last straw, and the FDA and other regulators now require basically that all anti-diabetic agents must have a CV outcomes trial. The aim of these is really to demonstrate safety and non-inferiority with respect to placebo, rather than demonstrating superiority, which of course is the best that one can expect and hope for. So this is the data effectively and the statements from the FDA, which resulted in removal of rosiglitazone from the market. Basically, the advisory committee concluded that rosiglitazone use was associated with a greater risk of MI than placebo, metformin, or sulfonylureas. There were some suggestions from subgroups of patients on long-term nitrate use or insulin use may be at high risk. As I mentioned earlier, 3 years later, when FDA reanalyzed these data using 52 trials instead of the 42 trials that were used here, they concluded, and the direct statement is that actually the results are inconclusive, therefore we're going to remove the restrictions, which to my mind is rather strange. But there we go. Okay, so pioglitazone. Pioglitazone had not actually been tarnished with the same profile that Rosie had, and in fact, it is probably this is probably the best proof in terms of the role of insulin resistance in disease and in terms of cardiovascular risk benefit. From a pioglitazone trial that again I think Bob is going to go in detail. This is the IRIS trial. Notably, this study was performed in insulin-resistant patients, but they were non-diabetic. So despite the fact that they are non-diabetic, treatment of insulin resistance is actually causing a better survival in these individuals, which is, if there was ever additional proof needed of the role of insulin resistance in determining cardiovascular health and risk involvement in disease. Okay, so message from the glitazones is that they have certainly compound-specific effects. The risk of CV, heart failure, bladder cancer, and impact on lipid profile, which I didn't show, varies between pio and rosie. Neither pioglitazone or rosiglitazone have liver toxicities that was associated with TROR. The reason that I would like to focus on this compound-specific effects is because in the literature there is always reference to glitazones are no good or DPP-4 inhibitors have low efficacy, and I think we need to be really careful at the patient level how we are addressing each compound and understand each compound in order to use it to the highest benefit for the patient at the end of the day. Now I mentioned I wanted to share with you some of my experience in the drug discovery area, and I was fortunate enough to join Boehringer Ingelheim some years ago when we were in the early phase of developing DPP-4 inhibitors, and in particular linagliptin. When the program was started, vildagliptin was very well advanced in the clinic, and there were certainly these two compounds also in various stages of development. To the chemist's eye, these have very similar structural moieties, and one of the goals of the program in Boehringer Ingelheim was actually to move away from these structures. This was not due to any prior knowledge about any sort of issues or disadvantages of these and advantages of this structure. We just wanted to be different with the hope that possibly there would be some kind of different profile. And the result of modifications of compounds like this resulted in this compound, which we used to call BI-1356 and now known as linagliptin, that is on the market. Through preclinical assessment in some of the models that Claire mentioned, in this particular study, Zucker fatty rats, we demonstrated that inhibition of the enzyme DPP-4 by this BI-1356 molecule resulted in elevations of GLP-1 levels in conscious moving rats undergoing normal feeding. And this was associated with a better insulin profile that resulted in a better glucose control. I'm not going to show you too much of that data, but suffice to say that when the compounds actually went into the clinic, it was identified that it was not being excreted via the kidney, unlike all these other DPP-4 inhibitors. But in fact, the route of excretion was mainly through biliary pathway. The result of this, of course, as we all know, diabetics also are at risk of kidney disease, and many of them have various stages of chronic kidney dysfunction. The result of this profile is that linagliptin does not require any dose adjustments, whereas all these other compounds require dose adjustment if the patient has significant kidney abnormalities. So in the same way as not all glitazones are the same, not all DPP-4 inhibitors are the same. Andrew already mentioned the risk of heart failure with saxagliptin, but as I mentioned, the dose adjustment story with respect to CKD patients also should help physicians decide which compound to use in which patient. Last but not least, and just to run quickly through this, SGLT2 inhibitors, these inhibit sodium glucose co-transporter in the kidney and result in, instead of reabsorption of the glucose back into the circulation, result in the excretion of excess glucose into the urine. This profile was already presented by Andrew very eloquently, so I'm not going to go through that, but just to say that although it does demonstrate a great set of data in terms of cardiovascular benefit to the patients, this data also raises some questions. Firstly, what is the mechanism of empagliflozin reducing cardiovascular disease? Which patients are best candidates for therapy? We are having increasing numbers of classes of compounds. How are we going to distinguish which patient is going to respond better to these versus anything else? And just as importantly, is this a class effect of SGLT2 inhibitors? All indications are that it probably is, but we have to wait for the data from outcomes data from the other trials of the other SGLT2 inhibitors before saying yes to that. So we now have a whole range of compounds for treatment of compound classes for treatment of diabetes, ranging from the SUs and glucosides, glucosidase inhibitors, TZDs, DPP-4 inhibitors, incretin mimetics, SGLT2 inhibitors. And whereas this brings a whole load of armories for the physician, I think it also generates a lot of questions. Each of them has advantages and disadvantages. And whereas the guidelines generally just talk about, well, look at the patient, decide whether they're overweight or not, and based on their phenotype, decide what to prescribe them. I think we would do well if we understood actually more about the mechanisms of each of these in order to try and see whether patients can be divided in various subpopulations, if you like, those who could benefit from one type of therapy versus another. And I think we're really at the very early stages of that understanding. So guidelines effectively pretty much look at medical history, physical examination, lab results, start with diet and exercise, put a patient of metformin, 3 months later, if they're not controlled, add one of these, 3 months later, if they're not controlled, add another one, and just carry on. With very limited scientific understanding as to why is this happening, why is some patient responding to one type of therapy versus another. So, as if it wasn't enough that we have all these different classes of molecules already, there is a lot of effort in order to develop some additional classes and in fact follow up to these existing classes. In terms of follow-up to existing classes, I think it is fair to say that there is increasing data, certainly from the GP studies, and other population studies, that basically adherence to therapy is inadequate in diabetic patients, probably in most chronic indications. And one of the efforts is basically try and help people to help patients in order to adhere to their medications better. So, for example, GLP-1 agonists that are injectables, there are now trials of oral versions of these GLP-1 agonists. There is effort in terms of in order to develop devices whereby you could fill these devices with compounds that would otherwise be injected, and this device would then be implanted in the patient for 1 year, and it would provide slow release of the therapeutic peptide. There are also additional mechanisms that are being explored. Claire just mentioned GPR40, there is and 120 agonists, 119 agonists, glucokinase activators, mitochondrial activators, and the list just goes on and on. And is not exhaustive. As if that wasn't enough, there are also companies looking at implanting bioelectronics devices. So there is recent data to show that inhibition of a carotid sinus nerve in insulin-resistant animals, diet-induced insulin-resistant animals, results in improvement of insulin sensitivity. This would be a device that potentially can be implanted on top of the sinus nerve, and via an external manipulation of a frequency of an electronic signal, you would manipulate the activity of this nerve by some totally unknown mechanism. This nerve is known to enter the NTS, but the subsequent mechanism is unknown, really. You could potentially impact glucose control. So this is an exciting development. Very, very early days. It would be several years before we see anything like this in the clinic, I would say. But nevertheless, that's one of the angles that has not been tackled by any of the other existing treatment strategies. So just to summarize, there's a range of therapeutic classes available to manage glucose control. CV outcome safety trials are really embedded in the development of anti-diabetic agents at the moment. Individual agents in different classes may show distinct properties, and I think physicians should really try and use this to the advantage of the patient in terms of their control. And CV outcome superiority trials, as will be discussed later, are ongoing. So there is certainly tremendous opportunity for individualizing treatment and going from this sort of current monotherapy, dual therapy, triple therapy approach to understanding, can we determine who is going to be a poor responder to one or the other type of therapy, and basically give them something that is going to respond. And as was mentioned earlier, is there a role for early combination treatments? Which combinations are going to be better for which patients? Thank you.